From Microbe TV. This is Office Hours for Wednesday, July 10th, 2024. I'm Vincent Racaniello, and welcome to your viral corner of the internet. That is the place where you can learn about viruses. Welcome, everyone. Here I'm in New Jersey tonight, where it is 29°C, 68% humidity. It's cooled off a bit. Previous days were really hotter and humid, more humid. Let's thank our mods before we proceed tonight. Thank you, Andrew from New Zealand, for coming by. Thank you, Frank from New York, for coming by. Thank you, Steph, San Francisco, for coming by. And Frank, we said Frank, sorry. And Tom from the Oregon Coast Range. Thanks, all of you, and Les from California. But he will miss, but he popped in to say hello. Thank you, Les. Appreciate it very much. Those are our moderators tonight. They keep the tone civilized, and this is my Twiv shirt. I can't figure out where to put my finger here. This is a long sleeve t-shirt from Cafe Press. I really like the long sleevers. And yes, I wear long sleeves all summer. I just don't like short sleeves. I hate the cut off at the arm. In any kind of shirt, I don't like it. Sorry. And tonight's agenda, by the way, I do have an agenda. I made a list. Tell us where you're from, and I will go through and say hello to everyone individually. I have a couple of questions for you and discussions, and then we're going to have a mini lecture. It's going to be all about why the polio eradication effort is a failure. Okay. If you're seeing glitches or whatever, it's not me. I have perfect internets here. I'm doing hardwired. We got gigabit stuff. Don't worry about it. It'll settle down. You know, when it gets hot. Okay. Steph is texting me. Let's see what Steph wants to tell me. Your internet is stuttering. Well, all I can tell you, it's not me. It's not me stuttering. I'm looking at the feed here on my laptop, and I don't see any stuttering. Sorry. I don't want to start and resume. I'm going to have to deal with it. Because I don't hear any stuttering. I don't see any stuttering on my end. I didn't change anything. The only thing is the internet, which I can't control, right? So maybe it'll settle down. Who knows? 8 p.m., busy time. Everybody turning on the tube and streaming, right? Let's see. I'll turn off my Dropbox. Yeah, we can do that. That might help a bit. Okay. So let's see who you're from, where you're from. Here we go. Wayne is from Urbana, Illinois. Thank you, Wayne. Good to see you. I know that Dagfinn is from Norway. There we go. 2 a.m., 17°C and raining. Well, there you go. It's a lot cooler there. Lise is here from sunny Columbus. Who else do we have? Jan is from Tucson, Arizona. Brian is from Montreal. Montreal, lots of rain today. As I said, Tom is one of our mods from the Oregon Coast Range. Rima, is from Iowa. Elizabeth is from West Virginia. As Andrew says, who's from New Zealand, by the way. Hit the like button so YouTube knows to recommend this to anyone looking for an interesting science show. Not only interesting, but correct. Kang is from Chicago. Frank is from Santa Barbara. Mr. Doreen is from Elgin, Illinois. And I called Mr. Doreen that name one night on a stream because, frankly, I had forgotten his real name, and he just liked it. So he's now Mr. Doreen officially. Questella is from somewhere in Ohio. Here's Frank from New York City. MK is from Eastern Massachusetts. Abdur Rahman is from Brooklyn, New York, where it's 26. A little bit cooler here. Yes, Rima is from Iowa, right? Sandra, I don't know where you're from. Doreen is from Western Massachusetts. Mike is from Seattle. Jamie is in the Toronto airport away from home. Jessica is the University of Toledo. Ariozone from Asheville, North Carolina. Garrett is in the Southern Oregon Mountains between Ashland and Klamath. Is it Klamath or Klamath? Klamath Falls. Claire is from the UK. Sorry about the glitches. Elena is from Reno, Nevada. Privyet. Privyet. Yes, maybe the Russian satellites are making me stutter. Checking in with Veronika. Maybe she'll come later and tell us. Logotic is 23 degrees in Chicago. MJ is from Lansdale, Pennsylvania. First time attending? Welcome. Great to have you. Hope you like it. This is an informal version of our podcasts with just me usually and occasionally a guest. But this is for you. This is for listeners to be interactive, right? A podcast, you can't really do that. And yes, I'd love to have more guests. And I will. It's, scheduling is a bit of work. So it's easy for me to sit down and just do it. Yeah, and not everyone can do it at 8 p.m. on a Wednesday, but I will have more guests, yes. Artemis 21C and 92% humidity in Montreal. Wow. So here it's 69% humidity and 29C. Wow, it's interesting. No, interesting. Spamlet is from Bedfordshire, UK. Hat is from LA, San Francisco Valley. A cool 103F in the shade. Doreen is from Western Massachusetts. Betty's from Washington Heights, New York City, my old stomping grounds where I still have an appointment at Columbia University Medical Center. I'm glad it's settled down. Rita is from rainy Michigan. Jefferson Space, Miami 92F. Sandra is in Mexico. Chris is in Montreal where it's raining. Privyet. And Jennifer is from Denton, Texas. Cute child there, Jennifer. Very nice. Alpha Crab is from Michigan. Well, that's very interesting. EHS News says at the beginning of the pandemic, I emailed the local TV anchor things I learned on TWIV, and she covered it on the local news. That's cool. I'm glad. I've heard stories of people telling people who tell people, and it propagates. What we may not have a billion listeners, but our information propagates, so that's fine. However, we can get it to work is fine. Let's see my agenda. Okay, we got where are you from? Second thing I wanted to ask: have you seen immune boosters? 2 have been published so far. Third one is going to be published tomorrow. Tomorrow is Thursday, right? Is it true? Is that right? Am I speaking the truth, or is it already published? Number 2. And next week is going to be the third one. I misspeak. But we've had 2 so far. Yes, this week's immune booster number 2 with Michael Tal, sex differences in immune responses. And then the previous week, we had Luis Montané from the Wistar Institute, from bench to bedside. The reason I ask is because this was an experiment. Immune boosters. There was a immunology conference in Chicago. Cindy and Brianne and Steph were going to go, and Karen said, "Why don't I go with them and we'll record some interviews? They can line up interviews." I said, "Yeah, go. I don't want to do that. It's not my thing. I mean, it is, but I didn't particularly want to go to that immunology conference." So I said, "Go," and they ended up recording like 15 or 12 interviews. The first one we released as an immune because it was long, and that was really good too, with Joseph Larkin III, T cells and tolerance, really great. And then the rest are going to be boosters, right? Clever name. So I just want to point it out. I like them. I think they're really good. They're short. And so the idea would be when people go to a meeting, we can send Karen. I don't have to go to every meeting. It's a virus meeting, I'll go, but not every virus meeting, right? And she can facilitate recording, right? So she did the recording in a hotel suite, and turns out good. Turns out well. So I like it very much. So please check them out. They're short, and I think very informative, and they bring out the personality of the person that they're interviewing. We typically just have 2 hosts interviewing one person, otherwise it gets too overwhelming. So they switch off, right? So I just wanted to point that out. This is the kind of thing we can do with your support, right? Because it costs money to send Karen and rent a hotel suite and have the equipment there, so forth. And then to edit it, we have to pay people to edit it. So with your support, we can do new things like this. I really like this idea of one-offs at meetings. By the way, you may say, why did you have to rent a hotel suite? Well, this meeting was in Chicago, and if you do anything at the venue of the meeting, I don't know where it was, convention center, whatever, you have to get union people to do your recording. And I'm not going to tell you how much per hour that is per person. Not that I don't want to pay them. They're talented people. It's great, but it's not what we can do. However, if you are in a hotel, you can interview anyone you want. There's no problem doing it in your room. So Karen got a suite, and there's a separate room where she could set up the interviews apart from where she was. So I like this very much. I hope you enjoy them, and we'll extend it to other meetings as well. All right, they're short for people who don't have a lot of time. There's the mix of people in the world, right? Some people have a lot of time, some people don't. It's fine. I understand. I don't have a problem with that. Okay, and then one more thing before our mini lecture. So Kang wrote me this week, and Kang last week had asked. So our mini lecture last week was therapeutic viruses, right? So you can modify viruses to treat diseases, to become vaccines, to cure cancers, and so forth. Boy, we got the branding going on here, don't we? Woohoo! cafepress.com/twiv, I think. Let's check it out. Yep. cafepress.com/twiv. Cool. Okay. And we we don't make a lot of money on these items. They're mainly for promotional purposes. We make $1 per item, okay? But there's all kinds of stuff besides clothing and drinkware and office supplies, blah, blah, blah. So check it out. Okay, back to Kang. He had said, I asked you about whether these therapeutic viruses were gain of function. And it was a great question. And he said, it evoked a little more anger than I would have liked. And I'm sorry if I seemed angry, but when I expect a lot from people who govern this country. And frankly, if you say we're going to ban gain of function research, that's nothing. That's just not doing your job. And I shouldn't have got mad, but I did because I'm very passionate about not just virology, but science in general. Science makes our lives what they are. I don't care if you like science or not. I don't care if you believe in it or not. I don't care what you think, because you're wrong if you think science doesn't contribute anything. It does. I mean, this technology for us streaming, that science, you staying healthy, vaccines, and microbials, antivirals, etc. It's all from science. And way more rockets, right? Satellites, your cars, the macadam on the street. Everything is from science, more than you think. So it bothers me when people are misinformed about science, especially people who should be. But I will try to keep it calmer in the future. I just want you to know that I am passionate about this. I believe in science. I believe it should be represented properly. And that's why we do Microbe TV. That's why we do all these science programs, because I want people to learn. We're trying all different kinds of formats, just haven't even come close to exploring all the formats. But that's because I want to reach as many people as possible. So it was a good question, Kang. And by the way, Kang suggested that I get some people on Twiv who do therapeutic viruses and have them explain what they do. And that will be a good way of showcasing gain of function research. So, yeah, that would be cool. I agree. And I will try and calm down. But if I don't, it's not really anger, it's just passion. Okay. And we have 115 people. That's pretty cool. 65 likes. So hit the like button right below the video window. Hit the like button. I mean, Sandra says, I believe in science too. I think everybody here believes in science. These are the converted that we're talking with here. And I appreciate that. And you can take what you learn and move forward. But we'd like to know that other people are reaching it. We're reaching other people as well. So anyway, I'm glad you like immune boosters. Let's see. We have a few other people who have joined us. I think the last person was Jennifer. I think we did Alpha Crab because I remember that name. And hello from Singapore. I don't know how to say the character, but hello. Wayne is in Urbana. It's wonderful. Welcome. Markle is from the east end of Long Island. Haven't seen you in a while. Welcome back. Bernard is in Hyattsville, Maryland tonight. Welcome. Marceline is from the Philippines. Welcome. It's pretty far away. Karen's playlist is from Drizzling, Maine. You appreciate my passion. Don't stop. I won't. I won't stop. I will try to moderate, but I always have this passion and defend science. Yes, Dagfin, we're here because we believe in science. Right. We have to get other people here that don't believe in science. Maybe not here, but you have to talk to them. And we have an example of that here coming up. Rip DeComp is in Taos, New Mexico. Welcome. And Jacob is from Texas, but is in Ann Arbor for a conference. John is from northern Thailand. The bottle of Listerine. Doreen writes, we respect that passion. Isn't passion what drives most great actions? I think so. But there's a fine line between passion and getting angry. And as Lagodic says, it's not really anger, it's just passion. I think so, but I come off as being angry. I'm not really, although in the case of the lawmakers, I'm a little angry at them that they say stupid things, but I could say that calmly. I'm very disappointed in you. Not that they care, right? They wouldn't care if I'm disappointed. Fire EMS is from Atlanta. People are still drizzling in. Let's see. Monir is from Bangladesh. Cool. That's very cool. Must be really hot there. Okay. We have some questions here, and we have more people. But I want to do a little mini lecture first, which will probably take a half hour. Then we'll get back to that. This is very This is very important. I want you to watch, and you can ask questions during it. I'll make an effort to look for the questions as they're coming up. All right, because sometimes it doesn't work if it's all the way at the end, right? I like this one. Maybe it's passion with a dash of frustration. That too. That can color your passion, right? It can make it sound like you are angry. It's a dividing line. But I think you get it, okay? I've explained myself. I wanted to do that, and I think you understand. Okay, let's share this screen. And the title of my talk is Polio Eradication Failure. Yes, it's a failure. I'm waiting to see it come up in the stream. There's about a 20 to 30 second delay. Can I change this? I don't want top chat. I want live chat. Not there we go. Everything. I always forget to do that. All right, so the title of this is Polio Eradication Failure. I want to explain the polio eradication program and why I think it's a failure. And it's not just me. Many other people think so as well. I'm not going to name them. But, one of the motivations for doing this is an opinion piece published in the New York Times back in May by Richard Conniff. The end of polio is in sight. What have we learned? Where is it? Where is this slideshow? I haven't seen it come up yet. I hope I'm showing it. I want to make sure before I talk too much. It looks like it's in the stream there. But do you see the slides, folks? Someone just say yes, slides or something like that, please. And I'll wait. Here's the delay between when I talk and when it shows up in the stream. There we go. Polio eradication failure. Okay. So in May, this opinion piece was published in the New York Times. The end of polio is in sight. What have we learned? So Richard Conniff is not a virologist. Richard Conniff, I think he writes for various places like NPR, writes about human biology. He doesn't understand the nuance of eradication. He's talking here about this initiative of eradication. It's a consortium, right? We have the Gates, we have Rotary International, WHO, CDC, and the Gavi, the Vaccine Alliance. He says, if the initiative succeeds, it will be the culmination of a campaign that has reduced the incidence of paralytic wild polio from 350,000 cases in 88 to just 12 known cases last year. Okay, so that's wild polio. It completely overlooks, and I'm not getting angry, folks. I'm just getting strident because Richard doesn't know his stuff. Why is he writing an opinion piece about polio eradication? And that's why I want to do this mini lecture. It's not just about wild poliovirus. We have vaccine-derived polioviruses circulating, and they are the threat, and they are poliovirus. It doesn't matter that they're vaccine-derived. They are still poliovirus. They still paralyze kids. So why are you saying, oh, this is great, we're going to eradicate wild polio, and totally ignore vaccine-derived polioviruses? Are you assuming, well, first of all, do you even know that they exist, Richard? And if so, do you understand why they're a problem? So Richard is not going to see this lecture, but you guys will. Here we go. First, a little background on poliovirus. This is the virus I spent my life working on. I started working on it in 1979, and I stopped in 2022. Many years, 40 plus years. It is an RNA virus with a plus-stranded RNA genome. So here's the capsid structure. As you can see, it's a pretty thin shell, and inside in purple here is the viral RNA. At least some people see slides, not everyone does. I'm sorry, they're up here. Let's do a little history. Poliovirus emerged as an epidemic disease at the beginning of the 20th century, but it's been known since 4,000 years. We have this Egyptian carving from 1500 BCE, which shows a man with a paralyzed leg, probably polio. But there were just sporadic cases until 1900s, early 1900s, when suddenly we saw outbreaks, became epidemic. So the epidemiology changed. So that's an interesting question, but the virus has always been around, yet it changed how it appeared in outbreaks in the early 1900s. And we now understand that this is because poliomyelitis, the disease caused by poliovirus, and I'll talk about the pathogenesis in a moment, is a disease of modern sanitation. What are you talking about, Racaniello? Before 1900, the virus circulated freely. Most babies are infected shortly after birth. Why is that? Well, poliovirus is ingested, it goes into your intestinal tract, it reproduces, and then you shed it in your feces. And before 1900, what did we do with feces? We threw them in the backyard or out on the street. You had a chamber pot in your room, you used it at night, in the morning you dumped it out. No, there weren't any sewers, there weren't any toilets. So there was poop everywhere, and babies got exposed to poop very early on. I mean, mothers are handling these chamber pots, or fathers, their hands get contaminated with polio. They pick up the baby, and it gets infected. So every baby pretty much got infected within the first few months after birth, but their mom had given them antibodies across the placenta, and those antibodies, which they had in their circulation, protected the babies from getting paralytic disease. So maternal antibodies prevented paralysis. But what happened in 1900? Yes, as Dixon would say, the greatest invention of humanity, the toilet and the sewer that now takes away the fecal matter from babies in their first months of life. Infections delayed till maybe you're 5, 6, 7, 8 years old. There are thousands of kids now that are susceptible to infection. They don't have maternal antibodies anymore. And the virus finds its way to them, and boom, you have outbreaks like we saw here. Tens of thousands of cases a year. That is the beginning of epidemic polio. And then it becomes notable because all of a sudden there are thousands of people getting paralyzed at once. So let's talk about the pathogenesis. That means how poliovirus causes disease. You ingest the virus with fecally contaminated material. I think you understand how that happens, right? You touch something that has fecal contamination, someone's hand who didn't wash their hand after going to the bathroom, and then at some point you put it in your mouth, which happens, and you get contaminated. Oh, let me turn myself on here. There's a way to, I've forgotten how to do this. Camera. I don't know if that's it. Let me see. I'm going to show my face here. No, that's no good. I put it above the screen share. Yeah, there it is. Just want you to see me because I get animated and you can't see me. I'm not sure it's going to show up, but we will try. I put it above the slides in the hierarchical layers, so I think my face will show up soon. All right. We're going to pathogenesis. We eat the virus, it goes into our intestines, it reproduces in the intestine. It crosses from the epithelial surfaces of the intestine through our lymph nodes into our blood and causes what we say is a viremia, virus in the blood. So lots of reproduction in the gut, virus in the blood, and in 99% of infections, that's it. It's a mild or asymptomatic infection. And then in a few weeks, it's over. And you're also shedding a lot of virus in feces, and that's how it's transmitted. In 1% of infections, the virus gets from the blood into the central nervous system. It kills neurons, and that makes your muscles paralyzed, and that's poliomyelitis. And there are 3 serotypes, that is, 3 antigenic types of poliovirus. In the wild, those 3 serotypes only infect primates, humans, mostly humans. There's some anecdotal. In wild, I would say only infect humans, although in the lab you can infect non-human primates. So let's change this to humans. All right, so that's the pathogenesis. So the disease itself, these are the reported paralytic polio cases and deaths in the U.S. since 1910. And so let's just look at the paralytic cases here on the y-axis, tens of thousands. You can see in the early 1900s, we start having these epidemic outbreaks. And get bigger and bigger and bigger. Then, of course, post-World War II, it gets huge because the baby boomers, right, the service people coming back from the war having kids, and those kids are additional hosts for poliomyelitis. And about then, FDR gets interested in polio. Now, FDR, of course, had polio in his 30s. He couldn't walk without assistance. You can see he has leg braces here. He used the crutches. He used wheelchair. Could not walk on his own. He was very frustrated, and he wanted no one else to be paralyzed. So he started the March of Dimes, the National Foundation, and raised money to develop 2 polio vaccines. By the way, in the 30s, 40s, 50s, hospitals were full of iron lungs. Look at this photograph of a hospital ward. Each of those iron lungs is a machine that breathes for you. You put in this machine and it applies pressure to your lungs, gentle pressure, and then the pressure is relieved and your lungs expand and your chest expands and you breathe in, right? So you see a little girl there. You can see their head sticking out. And many people got out of these eventually, like up here in the upper right. There are people who have no trouble breathing. But, there are a lot of kids in getting paralysis. So FDR developed a vaccine. So actually, he funded the development of 2 vaccines. One by Jonas Salk, the so-called inactivated polio vaccine, and one by Albert Sabin, oral polio vaccine, or OPV. So these are 2 acronyms we're going to hear a lot, IPV, OPV. IPV, inactivated, it's injected into your muscle. It's non-infectious virus that instead is protein mainly and RNA and gives you a nice immune response, whereas OPV, you drink it, it immunizes your intestines transiently, and it also gives you antibodies and T cells in the blood, as does IPV. Now, IPV was licensed in 1955 in the US. Its use led to the drop of cases to a few thousand a year. In 1962, OPV was licensed, and its use led to the elimination of wild poliovirus in the US. And in many other countries, had success using OPV to reduce the numbers of polio. So with that in mind, the WHO, the 41st World Health Assembly, right, the governing body of WHO, in 1988 published a document, WHA 41.28, you can look it up, Global Eradication of Poliomyelitis by the year 2000, okay? Global eradication of poliomyelitis. This was not a bad thing back then because we didn't know a lot. It seemed reasonable. But it was poliomyelitis, the disease, not the virus. But suddenly, along this timeline, here comes stop polio transmission. No, why did you put that in there? No, that's the big problem. That's what we're going to talk about today. So I say this is feature creep. It was originally poliomyelitis, then it became poliovirus 2, and that broke it. That broke the eradication program. Why is it? All right, I need to tell you a little bit of molecular biology of poliovirus. So Albert Sabin, remember, he made these 3 serotypes of OPV, which you drink, you get immunized. Those are mutants. He made those viruses, types 1, types 2, and type 3, by passaging the virus in different animals and different cell types until he found one that would not cause polio in experimental animals. It was tested in humans and licensed as the polio vaccine. Okay, so 3 serotypes of OPV, type 1, 2, and 3. In the 80s, multiple laboratories, my own, Phil Miner, Jeff Almond, wanted to know the mutations that caused these OPV strains to be less fit and therefore not cause paralysis. And there were a limited number of mutations in the genome. All the viruses had a 5' non-coding region mutation. That is a part of the genome that does not code for proteins. And others had coding region changes, amino acid changes. Yes, Sabin Sundays, exactly. Give everyone OPV on a Sunday. Here are the locations of these 5' non-coding regions, mutations in OPV. Here's the poliovirus genome from the 5' end to the 3' end. The 5' end has an untranslated region, about 740 bases. It doesn't code for any protein. And it's highly structured, right? As you can see here, stem loops. And stem loop 5 expanded on the right. Each of the 3 serotypes of poliovirus has a single base change in this stem loop structure. Here's the type 1, the type 2, and the type 3 serotype of OPV that reduce its fitness and makes the virus reproduce less in people. It doesn't get into the central nervous system. And that's how they work. And we did experiments in the 90s that illustrate this. Here's an experiment in mice where we infect them with 2 different viruses, one with a C, polioviruses, one with a U at 472 and one with a C. So the U is from the vaccine strain. The C is from the wild type parent. These viruses differ only at 1 base out of 7,440, just 1 base. And if you put that virus right in the brain of mice, the U-containing virus will never paralyze mice. It does not paralyze them, whereas the C-containing virus does. And the reason is the C-containing virus reproduces in the mouse brain. This is a growth curve of these 2 viruses in the mouse brain. So we're looking at plaque-forming units per gram on the y-axis and a day post-infection on the x-axis. You can see the U-containing viruses are cleared. The C-containing viruses reproduce. And so the C-containing viruses, 1 base change prevents them from causing disease. It's really, really remarkable. But that is also the problem with OPV. And so in the U.S., starting in 1961, OPV was licensed. It replaced IPV. And this is a map or a graph of the cases of wild polio from 61 to 2003. And on the y-axis, the number of paralytic cases. So these are the total number of paralytic cases here. And you can see the number coming down after the introduction of OPV. But then you see these bars. And those are cases of polio caused by the vaccine. They're called vaccine-associated paralytic polio, or VAP, We didn't know this until many years later that these were actually caused by the vaccine. But you can see 10 to 12 or more a year. And eventually, all the cases of polio in the U.S. were caused by the vaccine from 79 onwards. 79 was the last case of wild polio in the U.S. until recently. And that's why in 2000 we switched back to IPV. And no more vaccine-associated paralytic polio because IPV is inactivated. It does not infect you, whereas OPV does. So this is the problem with OPV. It can cause polio. All right, so now the WHO is using OPV in its eradication campaign. So how's that going? Remember, 1988, they said we're going to eradicate polio by 2000. Well, now these are cases for 2023, previous 12 months. And actually, wild type 2 and wild type 3 were declared eradicated by WHO. So we don't see any more cases of paralytic polio caused by type 2 or type 3. We do see cases of polio caused by wild type 1 in Pakistan and Afghanistan. And these are very problematic because many places there cannot be vaccinated. There's extensive circulation of the wild type 1 virus. It has spread to other countries and caused outbreaks. So, that opinion piece, they talk about getting rid of type 1, wild type 1. Not so easy. Not so easy. But the real problem, and actually there are cases of vaccine-derived polio type 1. Those are the orange cases here. The real problem is type 2 vaccine-derived poliovirus cases. Those are all the green dots here throughout Africa and other countries as well, and New York in 2022. These are cases caused by OPV type 2 that has been in circulation in the population for some time. So it's VAP. So the reason this happened is because in 2016, WHO decided to withdraw OPV2 from trivalent OPV. So remember, OPV has types 1, 2, and 3 in it. They said in 2016, type 2, wild type 2 is eradicated. So, and we're only seeing vaccine-derived type 2. So why don't we take the type 2 vaccine out of the OPV so that it won't cause this problem anymore, okay? And WHO, the CDC said we've done modeling and it's okay. It will be okay. Well, it's not okay. It's a big error and we're paying for it to this day. And I'll tell you why. So first of all, after the OPV withdrawal, the number of poliovirus type 2 isolates declined, right? And here is 2016 and 2017. And these are number of poliovirus type 2 isolates on the y-axis. There's some sewage samples here, and then there's samples from patients with paralysis here. And you can see by 2017, looked like OPV2 was gone. It was no longer, this VAP, type 2 VAP was no longer circulating. So, okay, looks like the withdrawal was a good thing. No. Turned out there was wild polio in the northeastern part of Nigeria, the Borno state, that had been undetected because there was no environmental surveillance. If you don't look for poliovirus, you're not going to find it. In July of 2016, there were 3 confirmed polio cases in Borno state, and circulating VDPV2 was also found in this area. Now remember, here they didn't find it because they couldn't look in this part of Nigeria. So what did they do? WHO said, let's go in with monovalent OPV2. So that's just OPV2 on its own without types 1 and 3. And then that reintroduced OPV2 into a population that was now under-vaccinated against type 2, right? Hadn't been vaccinated with type 2 for a while. So what happens? Boom. CVDPV2 circulates again. So here's 2016 withdrawal, 2017. What we're looking at here, cases of paralysis. We're looking at environmental samples of poliovirus and others. And 2019. So 2017 was the year of the Boko Haram. 2016, 2017, the reintroduction of OPV2, 2019, 2020, 2021, lots of circulating vaccine-derived polio too. So the idea of withdrawing OPV2 from the trivalent was a disaster. It was wrong. Their modeling was wrong. And that's another thing I'm very passionate about. You know, if you make a conclusion based on a model, which people did all the time during COVID, it's only as good as the data you put in. And they didn't put in data that there was undetected type 2 circulating in Nigeria. How would they do that? They couldn't. They didn't know about it. So here is the total effect of circulating vaccine-derived poliovirus 2 globally. Since 2018, 3,000 different outbreaks, each of them by this dot, and each outbreak is more than one case. But most of the paralytic polio is caused now by circulating vaccine-derived poliovirus type 2. And for Richard in his op-ed to say, we only have 12 cases of type 1, we're almost there, ignores all of this. It's just not right. It's either not right or it's ignorance. So one of the lessons is that environmental surveillance, well, it isn't so difficult. You have to do it. If you don't look in the northeast corner of Nigeria, you're not going to know that there's circulating viruses there. Now, to be fair, the people who did the surveillance couldn't get in because terrorist group was controlling the area. There are many parts of the world that are not subject to environmental surveillance, which means looking for poliovirus in environmental materials, right? So this is a big problem, which we'll come back to in a moment. Now, then in 2022, we had a case of paralytic disease, poliomyelitis, in Rockland County, New York. It was caused by vaccine-derived Sabin OPV2 that had been circulating, and it was brought in from probably Israel. The CDC, the WHO, never thought that the virus could travel this far. CDC never looked for environmental polio in the U.S. because they said we're immunized and this is not going to come from anywhere else. Wrong. This individual had not been vaccinated. The virus is a typical strain of CVDPV2. It's a recombinant between OPV2 and Coxsackie virus A7. Was brought into the U.S. by an infected individual, and it transmitted until it reached a non-immune individual, which was this person. Now, the whole U.S. is immunized with IPV. It doesn't prevent fecal-oral transmission. In many places of the U.S., there is poliovirus circulating, but we don't see it because most people are immunized, so we don't see the paralysis. And VAP caused by circulating VDPV2 is about 1 in 2,000 people infected. So this individual was one of 2,000 people who were infected at that time. So the CDC decided to do some environmental wastewater testing for poliovirus that year. And this is a map of New York State and Long Island here and near Manhattan right here. And you can see these blue areas are samples that are poliovirus positive. And furthermore, they sequenced them and they could say, yeah, it's the same virus that paralyzed the Rockland case, the Rockland County patient. So this is circulation in the New York metropolitan area persisting over months of CVDPV2. And since then, the CDC hasn't released any information about this. So they don't want you to know that poliovirus is still circulating. Now, why did this individual get polio? Well, the vaccination rates in Rockland County in some places are horrible. This is by zip code. And you can see this is by age 2 years. Most people are 70 to 90% or 90 to 100, but these particular zip codes 0 to 50%, 50 to 80%, not good enough. 50 to 55%. That's why he was infected, because he hadn't been vaccinated. Same thing with Orange County. Lots of counties where people don't get vaccinated against many infectious diseases. And there you go. So how do we eliminate circulating vaccine-derived polio 2? I hope I've at least taught you that this virus is circulating like gangbusters all over the world. It's in the U.S., it's in Africa, it's in many other countries. Many countries use OPV. That's what the WHO gives them to use. And now we have a huge type 2 immunity gap because we haven't immunized anybody except in outbreak situations since 2016. And so that's why we have all of these cases in places where people are no longer immune. Kids are no longer immune to OPV2. And it's not just Africa. You can see here, the Arabian Peninsula, here, Afghanistan and Pakistan, where immunization rates are low. But also, they're just not getting OPV2. It's taken out of the vaccine. Good move, folks. Okay, so what do you do? Well, a couple of years ago, more than a couple of years ago, a group of researchers got together and said, let's take OPV, the live attenuated polio vaccine, and engineer it to prevent this reversion in the 5' end. So we will get rid of vaccine associated polio. We'll use this vaccine. So they introduced a number of changes throughout the poliovirus genome. And these are the authors that were involved in this big consortium. They introduced mutations into the 5' end to try and prevent that U from changing back to a C. They changed the polymerase to make it less mutagenic and less recombinogenic. They did a few other things as well. Okay, so this vaccine was tested. It's called NOPV2, type 2 specifically. And look at the title, to prevent reversion to virulence. Did it? Okay, this vaccine went through phase 1, 2, and 3, given to hundreds of kids. And then 6 kids in DRC, 1 in Burundi, were paralyzed after given NOPV2. The viruses that paralyzed them are recombinants, where all of those changes so painstakingly introduced into NOPV2 have been removed by recombination with an enterovirus circulating in the gut of these children. So this is what the viruses look like. All that's left of type 2 is the capsid coding region. The 5' end is now from an enterovirus in the gut, and the rest of the genome is from an enterovirus in the gut. In other words, all those changes that were put in NOPV2 to stabilize it are gone. 2 recombination events, they're gone. So this virus is not the solution. It's a slight improvement over OPV2 in terms of vaccine-associated paralysis, but it's no different in preventing or stopping outbreaks than monovalent OPV2. And this cost Gates Foundation about $3 billion to build. It's a failure. It is a complete failure. It paralyzes kids. Any vaccine that paralyzes kids is a failure. I'm sorry, that's just the way I feel. So what are the lessons? Well, first of all, for poliovirus, attenuated infectious vaccines, forget it. Not a game at all, because these viruses can recombine with enteroviruses in your gut and they'll lose all the changes that you put into those genomes. And unless you do mass campaigns, Sabin Sundays, there will always be people who are not immune and they'll get vaccine-associated paralysis. So here, look, this article in Science in May. One of the authors of the withdrawal says it was an unqualified failure. The decision to switch polio vaccines in 2016, which in other words means to withdraw the type 2 component, was a failure. I've just showed you why it's a failure, because all of a sudden, after we tried to mop up that outbreak in Nigeria, and OPV2 came back because there was now a big immunity gap from not using OPV2. So now we're screwed because we have all these cases of type 2. And what do we do about them? Well, WHO thinks we can just keep immunizing with NOPV2 and they'll go away. But the more you immunize with NOPV2, the more you keep introducing NOPV2 into the environment and will keep circulating. It's not a solution. It's being stubborn. We need new polio vaccines. So the beauty of OPV is that you can drink it. It's really easy to give kids. So we need a new vaccine that could be easily administered, no injection, but does not cause polio. We need a new vaccine. How do you do that? We need research. However, next year, nobody can do research on polio anymore, or most people can't, because the bureaucrats have decided we don't need polio research, so you can't work on polio. As of next June, 2025, most places can't work on polio because it's considered a risk. And I don't get this, because the lab is no longer a risk. Poliovirus is circulating in the environment. How could the lab be a risk for release of poliovirus into the environment if it's already in the environment. These circulating vaccine-derived polioviruses are virulent. They cause polio. The lab is not a risk. Let people do research. This is really, really unfortunate. So what do we do? Should we use IPV? Well, many countries are, right? The US, it works really well unless you're in a place that doesn't get immunized, which is dumb, flat out dumb to not get immunized, okay? Many countries in Europe, Scandinavian countries, that all use IPV. It doesn't cause polio. You don't see any poliomyelitis. It's just the OPV being distributed by WHO in the eradication campaign. So could we switch to IPV? Well, in fact, WHO back in 2016, this is a little article from MMWR, they said introduce IPV worldwide. And they still to this day say, yeah, we should all be using IPV, but it's hard. So I think we could do that, but a lot of people are against it. Now, remember, IPV will not protect the gut against infection. So if the whole world used IPV, poliovirus would continue to circulate. All these vaccine-derived viruses, presumably wild type 1 as well, they'd all circulate, but if you kept immunizing everyone, it wouldn't be a problem. The issue is, could you immunize every country in the world all the time with IPV? It might be hard because the whole eradication program was sold by WHO to countries saying, well, you'd only have to do this for 10 years or 12 years. And then you can stop. But that's obviously not happening. So if we use IPV, no poliovirus eradication. IPV doesn't protect the gut from infection. Let me show you an experiment that proves that. I'm almost done. When they were testing NOPV2 in people, they fed it to people, and then they took their stool and looked for poliovirus over days, study days from 0 to 100 days, okay? These individuals had previously been vaccinated with IPV. And so, look, this is PCR positivity in red. Let's just look at that because there's also culture positivity. Look, despite having gotten IPV, they're shedding this NOPV2 for many days, up to 90 days in some people. So the red is actually PCR positive, culture positive. Culture positive means infectious virus out to day 90, people who got IPV. IPV does not protect the gut from infection. That's the point. You're going to have polioviruses circulating globally. A couple of other thoughts. The OPVs that Sabin made, we always used to think that those mutations specifically affected the virus's ability to reproduce in the central nervous system, but it's not. It just reduces their fitness. Less reproduction in the intestine, less viremia, less likely the viruses invade the CNS. Why do I say that now? Because the recombination with any gut enterovirus just restores fitness of the NOPV or OPV, and it replicates better. And so there's strong selection for reacquisition of fitness in the intestine. You can revert, although the NOPV2s don't revert in the 5' end. That's pretty cool. But they recombine like crazy, and that makes them more fit. Higher fitness, more reproduction, neuroinvasion. Okay, can we eradicate polio? No, not virus. We've replaced wild type with vaccine-derived polio. It's still polio. It still causes paralysis, acute flaccid myelitis. Neither IPV nor OPV prevent infection, even if the world, as I said, switches to IPV. OPV-derived viruses will circulate. Circulating vaccine-derived viruses cause many asymptomatic infections. And without wastewater surveillance, we don't do it here in the US, we don't know where it is. In fact, the Global Polio Eradication Initiative and WHO have recently called for increased surveillance of wastewater, but the US is not interested in doing wastewater surveillance for poliovirus. They say, we're immunizing with IPV. We're not going to get paralytic disease. We don't care if it's here. All right. So can we eradicate poliomyelitis? Let's fix myelitis. We need an I there. And can we eradicate poliovirus? All right. So we did eradicate smallpox, right? And these are some of the features that we think are important for eradication. We actually eradicated smallpox, the disease, as well as smallpox virus. So, virus reproduction only in one host, humans. Yes, for polio and smallpox. Check. Vaccination induces lifelong protection from the development of severe disease. That's yes for smallpox. Not really clear for polio, but long term, but maybe not lifelong. Asymptomatic spread does not contribute to disease transmission. That's the case for smallpox, but certainly not the case for poliovirus. Many, many cases of asymptomatic infections, and those people are spreading. Virus shedding occurs after the onset of symptoms. Yes, for smallpox. So you get the rash and then you start shedding. So people without rash are not shedding pre-symptomatically. But for polio, they shed before the onset of symptoms. Is severe disease caused by only one virus? Yes, for smallpox. No, multiple for poliovirus, including vaccine-derived polioviruses. So I think you can never eradicate poliovirus. Maybe if you immunized everyone globally at once with IPV and kept it up forever, you could eliminate the disease, but I don't think that can ever happen. I don't think the world has a stomach for it. And here's the real problem in the end. 99% of polio-infected people are asymptomatic, so we can't just look for paralysis, acute flaccid paralysis, to know where the virus is located. We need to do environmental surveillance, and we can't look everywhere. And finally, a virologist said to me recently, the polio eradication campaign is a failure because it's being run by epidemiologists, not virologists. The epidemiologists who do not understand the nuances of poliovirus biology, right? And just one simple fact that, where's my camera? Streamer. All right, there we go. The one simple fact that there's so many inapparent infections of polio is unknown to these epidemiologists. So that's why polio eradication will never happen. If you ever see an article saying, this is great, we're almost there, it is BS. And I'm not angry. I'm just passionate because, again, we have people making statements who don't understand because they don't know the facts. They don't know what they're talking about. Now, I know you have a lot of questions. So let's go back and talk about this. Okay, let's see here. Hello, Vanity Nutrition. Thanks for joining us. One of our moderators. What about writing to CONIF to tell them about vaccine-derived polio? Yes, I'm going to write a nice email to CONIF saying you are uninformed. Nah, try not to say that. To say it in the first sentence probably off-putting because that's what people do to me. I'll say you left vaccine-derived polioviruses out of the equation. You cannot do that. Oh my gosh. Good idea. Yes. I guess by doing this lecture, you are still working on polio. No, I'm not. I stopped working in 2022. And I couldn't be working anymore on polio, even if I had a lab, because of this containment thing where they don't want you working on polio because they think it's a danger for the environment, which is stupid, as I told you. Sporads. I don't know what a sporad is. Did I say that? I don't know. So sanitation was a bad thing as far as polio prevention. Yes, it was a bad thing because it delayed infection till the time when you no longer had maternal antibodies. A couple of months, right? 6 months or so. But so what do you do? Yeah, it's interesting because throughout history, for thousands of years, we had poor sanitation and we had sporadic polio. Then we improved sanitation, we get epidemic polio. So we made vaccines to take care of it. I think it's a reasonable trade-off. I think sanitation is better because you get other diseases from poor sanitation as well. All right. There's a comment here about India, but I don't want to talk about it because I don't know if it's right, and it's a little bit insulting. Gerald Ferraro, G. Ferraro. I hope you understand these vaccines now. IPV is what we're using in the US. It's injected, it gives you antibodies and T cells to polio, prevents disease, it doesn't prevent infection. We don't use OPV any longer in the US. By the way, Sabin OPV has been delicensed in the US. We are not allowed to use it. Yet, WHO thinks it's fine for us to use it in Africa and other countries. I think that's just a conflict. I don't think that's right. If it's not good enough for the US, it should not be good enough for any other country. I don't want any kid paralyzed. To which someone said, Vincent, don't let perfection be the enemy of good. Okay, well, we have IPV, which we know will not give you polio. So why don't we just use that? Yeah, it can be done. We immunized a lot of people with injected COVID vaccines, right? So don't tell me the injection is an issue. No, I won't buy it. Elise writes, perhaps you or a colleague could write a counter opinion. I pulled up the article and lots of comments that disagree the end of polio insight. Yeah, at least people understand. I don't understand why they let people write these commentaries who know nothing about what they're writing. That includes Lena Chan about the origin of SARS-CoV-2. Unbelievable. So it should be. Someone should write a counter opinion. I mean, it's not opinion, it's facts, right? I would state the facts. I'm not sure that I can do it. That's right, Donald. Ricky remembers taking the oral vaccine on the sugar cube. Yes, I did. I remember doing that. Can you use Salk and Sabin with some delay? So, yes, actually, that's another approach. You give a dose of IPV and then Sabin, and that prevents vaccine-associated paralysis. But it makes it more complicated. And by giving OPV, then you keep reintroducing those OPV viruses into the environment. I would rather, not do that and maybe hope that eventually these circulating OPVs, if we don't use OPV anymore, will go away. Why Sabin Sundays? So Sabin arranged mass immunization events in the U.S. on Sundays, and well, I think one of the ideas was they got a lot of people at churches on Sundays, right? So distributed the vaccines on churches. It was just a sugar cube, right? So it's easy to give. John asks, does washing with soap and water destroy polio, flu, cholera, and other viruses? So cholera is a bacteria. So soap will not destroy polio. It will inactivate influenza virus. But if you wash your hands long enough with soap, you will wash off most of the polio virus and then rinse them well. So you should wash them for the alphabet, right? A to Z. Long time. Visto asks, do we know why poliomyelitis came in waves? Yeah, I think the simplest explanation is that you have an epidemic which immunizes the population, and then you need to wait for new children to be born and become immune and build up the number of susceptibles that you need for an outbreak. It's a very typical enterovirus infection cycle. You have an outbreak, you immunize a good fraction of the population, and then you have to wait for more susceptibles to appear. Do I plan to get the new COVID vaccine? The latest variant. Yeah, which has been, no, I don't. I believe that I've had sufficient and to prevent severe disease. And if I got COVID, I would take Paxlovid. Sugar Cube was given on Sundays in Ohio. Biggest turnout in Cuyahoga County. Wow, you remember. Logotic remembers getting the oral poliovirus on Sunday in a sugar cube. Some years later, that's also how I took my first dose of LSD. Okay, well, hope they were both good experiences. About how long after OPV administration would the recipient develop signs and symptoms? So it has to be close enough so that there's an epidemiological connection, right? So you get paralyzed within a couple of weeks after getting OPV. It's very likely that that was the culprit, and they take a stool sample and they can show that you're shedding OPV-derived viruses. But of course, remember, you can get OPV, you can get polio from circulating OPV, right? Circulating vaccine-derived poliovirus, CVDPVs, CVDPP2, for example. And that appears just like a normal polio. You get infected, and a week or two later, you could get paralyzed. Tom, polio elimination goal was the feature creep driven in part by success with eliminating smallpox virus, driving optimism and perhaps hubris. I think that might be part of it. I don't actually know. But at the time, we did not appreciate the number of asymptomatic infections that polio causes, and we did not appreciate vaccine reversion, the extent of it. We had no clue how virulent those viruses were, virulent in meaning that they can cause paralysis, how transmissible they were. We had no idea about that. So I think it was a little bit of uninformed conclusion that led to that. I once asked D.A. Henderson, the architect of the smallpox eradication campaign, in an interview, which you can find on YouTube. Just search for Henderson Racaniello. Can we eradicate polio? He said, not the way we're doing it now. But he's no longer around to ask him about that. Melts in your mind, not in your mouth. That must be the LSD sugar cube, right? How does it revert to the wild serotype? The 2022 case calls the Fury. Well, so the virus that came into the U.S. had already reverted. It was a OPV2 strain that had reproduced in someone, I believe in Israel, and there it reverted by recombining. So there were two things. The 5' end reverted, and then the 3' end recombined with a gut enterovirus, and that made it more fit, and now it's circulating and causing paralysis. You know, if that person had been immunized, he would never have been paralyzed. It's just that the community doesn't believe in vaccination, and so this is the outcome. Markle says, I'm not sure I got the sugar cube. It was so long ago. Not sure if it was given at the local public elementary school. Wish I had a clearer memory. My mom never kept a vaccine card for us. Well, I have to tell you, I remember getting it at the sugar cube at the local elementary school, and my mom saved my vaccine card, which I have, and actually I have published on my blog. Wonder if I could find it. Let's see. Virology blog vaccine card. Let's see if it's there. Yeah, it is. Look at that. Holy crap. I'm going to share this with you. So this is my virology blog from March 2009. This is my vaccine record. So this is the school I went to, Edith A. Bogert School. And I got, back then they were doing monovalent OPV. So I got type 1 on September 8th, 63, type 2 on May 27th, 64, and type 3 on April 15th, 64. See? 3 serotypes on a sugar cube. I remember that. Yeah. But, I had already received, IPV in the 50s because I was born in 53. So I got both. I don't remember getting IPV. I would have been, a couple of years old. I don't remember that. Artemis models GIGO, garbage in, garbage out. Absolutely. If you eliminate something, it's like the models, they did Bayesian statistical analyses of the likelihood that SARS-CoV-2 came from the lab or the market. And some of them decided with high significance that it came from the lab. Well, what did you put in? Did you put in all the market data? Did you put in the fact that every human virus has come from animals? Probably not. You just biased it to get the answer you want. Unbelievable. With respect to Nigeria, went from a few circulating type 2 viruses to a lot circulating after monovalent vaccine with type 2 was given. All right, so in 2016, we stopped immunizing kids with type 2, right? We take out type 2 from the trivalent. We give them bivalent, which is types 1 and 3. So now for several birth cohorts, we have kids being born who are not immune to type 2 anymore. 2017, we have this outbreak detected in the Northeast. WHO decides to go in and immunize kids with monovalent type 2, which then is shed and starts to spread throughout the population across that province into the rest of Nigeria, where kids are now susceptible to type 2 because they haven't been immunized. And throughout in Africa subsequently, because now we have an immune type 2 immune gap because of the withdrawal of the type 2 vaccine. Big disaster. That's why that one slide in Science article, disaster, huge error to withdraw the type 2 vaccine. It's easy for me to criticize in retrospect, but I wouldn't have depended on just a model to say it's going to work. I would have said, in fact, back then I said, you might want to wait a bit longer. I think part of the modeling was based on the assumption that levels of circulating vaccine-derived type 2 were declining. So why not let them decline more on their own? Why do you have to take this drastic step of withdrawing the type 2? Oh my gosh. Ricky, how do you attenuate a virus and how does an adjuvant work? Oh, two big questions. So in the old days, Albert Sabin would attenuate poliovirus by taking the virus and growing it in different cells in different animals, with the idea that if you got it to grow better in other cells, non-human cells and non-human animals, it would lose the ability to grow well in humans. So he reduced the fitness of the virus for people. And so he would periodically test his viruses in animals and say, yeah, they're getting less and less able to cause paralysis. So that's kind of an empirical way of attenuating a virus. Nowadays, we can do genetic engineering, right? So for example, one of the new dengue vaccines, they removed 30 bases from the 3' end of the viral RNA. And it turns out that doesn't affect replication, but it makes the virus not cause disease. So you can be smart about it nowadays, or smarter. How does an adjuvant work? So adjuvants are chemicals mixed with vaccines to mainly induce inflammation. Because when you inject an immunogen into you, if it's an inactivated virus, it doesn't replicate, so it's not going to cause cell damage and it doesn't cause inflammation. And you need inflammation to make a really good immune response. So you mix adjuvants in that cause inflammation. If you've ever had an adjuvanted virus like Shingrix, really hurts after a few day or so after you get it because the vaccine is inducing inflammation. It really hurts. So that's what that is in a nutshell. Dramatic actions were taken during polio epidemics, closing theaters, swimming pools. These were all after the fact of infection. a legion of grandmas yelling at kids to wash your hands after using the toilet would be better. The other thing they did was to look for paralyzed kids and put them in a hospital, quarantine them, which failed. I mean, can you imagine? They went from apartment to apartment. If they saw paralyzed kids, they would take the kid and bring it to a hospital. But 1% of infections are the paralyzed kids. The 99% are not paralyzed. They're shedding virus. They're the ones who are transmitting it. So that was a matter of ignorance. We didn't know that until later. Does poliovirus have a fat section? No, there's no lipid. There's no envelope on poliovirus. Artemis says, how complicated would it be to reintroduce type 2 in OPV? So what WHO is currently planning, here's the brilliant thought. Remember, we talked about making NOPV2, right? A new and improved NOPV2, which is not really, but they're going to, this is crazy, take out the capsid region of type 2 from that virus and stick in the capsid region of type 1 and separately the capsid region of type 3. So they're going to have NOP1, 2, and 3. And then that's the idea. They'll give it to kids. But now, so type 2, vaccine-derived type 2 is the most transmissible. And now you're taking the backbone of that type 2 and sticking the type 1 and type 3 capsid on it to make 1 and 3 even more transmissible than they already are. Oh my God, this is so ill-advised. But that's the plan. This is really an S show, if you know what I mean. It's insane. There you go. This is insane. Yes, it's insane. Vista says you can drink the virus but not study it in a secure lab. Yep. It's another weirdness, right? I cannot study poliovirus in my lab because there is a risk for me letting it out and getting into the environment. But there's already polio in the environment, right? So it doesn't matter. A number of us are in the process of writing a letter to WHOs or try and get them to stop this containment deadline, but I doubt it will work, but we'll also try and publish it elsewhere. There's no way to prevent polio infection using IPV. No. Why use it? Because it prevents paralysis, right? It prevents paralysis. It doesn't cause polio. OPV causes polio. That's the problem. It causes polio. It circulates and remains a threat. You can't eradicate polio if you keep using a vaccine that's infectious, that circulates. Now, the WHO plan is to, someday in the future, have no more cases of polio in the world, and then they're just going to stop vaccinating. And I guarantee there will be outbreaks, because if you just look for cases, you're not going to know what viruses are there. But in fact, they will never have a time when there's no cases of polio, because these vaccine-derived viruses are going to keep circulating and causing paralysis forever. Thank you, Rob, for your contribution to science communication. Long live Microbe TV. Is there anyone with group with a natural genetic immunity? I was exposed repeatedly to measles and never contracted it. Yes, there are people who don't get infected with, or I should say, don't develop severe disease or any disease at all after being infected with certain viruses. And there are genetic reasons for that. And many people study that, like Laurent Casanova at Rockefeller University. And you can identify mutations in people. For example, if you want to know who gets severe disease, you can find mutations in interferon genes that confer severe disease. But resistance is harder. Yeah, because proving it and finding the right people for your study is very hard. So Marco says, okay, it still infects the gut, and that is the problem. No, IPV does not infect the gut. IPV is inactivated. When you are immunized with IPV, poliovirus in the environment will still infect your gut. You will shed it, but you won't get paralysis, and no one around you will get paralysis if they are immunized. So the key to preventing polio in an IPV-only world is just to make sure over 95% of people are immunized with IPV, which may be hard to do indefinitely in many countries. But I can guarantee you that the US and many parts of Europe and Scandinavia, they're going to keep using IPV forever. No way they're stopping. Are you crazy? You stop IPV tomorrow, then you have a couple of birth cohorts who don't get vaccinated, and boom, they're going to get, as soon as their maternal antibodies are gone, they're going to get polio because of the virus that's circulating. The circulating vaccine-derived poliovirus is in the environment that in the US we don't know about because we don't look for. Ay, ay, ay. Hand washing. What is the question? Hand washing is not a substitute for vaccination because there's always a break in the chain, right? Tom writes, recombination in positive strand RNA viruses is fascinating. The molecular mechanism driving evolution recombination amongst various enteros was a surprise. It's really amazing that, Saban develops this oral vaccine. He had no clue that in the gut of everyone are enteroviruses that are there all the time replicating. They don't make you sick. They recombine with OPV and they change its properties. Oh my gosh, who would have known, right? But that's why you study this stuff. And that's why you shouldn't stop studying polio because you never know what you're going to learn. And I'm telling you, none of the other enteroviruses are as interesting as polio and are as amenable to study. You can really learn a lot. All right, now Kang says, are there genes that make a virus neurotropic? So I used to think so for polio because I studied polio. I thought there are changes in the 5' UTR, right? One base changes that make the virus not cause paralysis. It must be a neurotropic determinant. It's not, because you can take this and substitute the whole 5' N with some other enterovirus that doesn't even have the same sequence, and that virus will cause paralysis because it's all about fitness. So a change from C to U in the 5' UTR of the Sabin type 3 vaccine strain lowers the fitness of the virus. It reproduces to lower levels in the gut. There's lower viremia. There's lower chance that it invades the CNS. But there's tremendous selection for revertants that arise in the gut that go from U to C. Why is there tremendous selection? Because they're more fit and they outcompete the other virus, the virus with a C. They outgrow them. So every kid that gets OPV, within a few days, they are shedding C-containing virus. Apparently, they're protected, most are protected against paralysis because by then you've already made some interferon responses. But it is very powerful selection for higher fitness. It's not neurovirulence. I mean, I can say that for poliovirus. I bet it's the same for other viruses. I bet it's all about fitness. It has nothing to do with specific neurovirulence, neurotropic determinants. Am I the only person with natural immunity to smallpox? I don't know. How do you know you have natural immunity to smallpox? I'm curious to know the answer. But if you never got it, how did you know that it was around, that you might have gotten it? And if you are, I bet there are other people out there as well. There always are. Don't tell Khan if he's uninformed or doesn't understand. Yeah, I know. I just want to, though, because it drives me nuts. But yes, I realize the way to get to him is not to tell him. He is not uninformed. I sit around thinking, dear Mr. Conniff, I'm writing to tell you how horribly misinformed you are about polio eradication. No, I know. He'll stop reading it. I will be calm. I will say, I would like you to know some information about polio and reversion that you may not be aware of. Ay, ay, ay. Where was the poop in India? Yeah, so John Brigg made a comment about poop in India. You can go see it. I'm not sure what I mean, it involves removing poop with your hand. So that's a good way to spread viruses, obviously. How structure of RNA is maintained, preserved. Well, the stem loops, right? They have base pairs that are on either side of the stem, for example. And if the stem is important, those base pairings will be changed. So if you make a change that on one side that disrupts the base pair, if it's important, you will get a reversion on the other side that now makes a base pair, if in fact that base pairing in the whole stem loop is important. So they're evolutionarily maintained. If you see these stem loops and lineages of viruses, you know they're important because otherwise they wouldn't be maintained. That's right. Hmm. Can you be an advisor to Congress to set the bureaucrats straight? I don't think they want me. I'm too opinionated about it. No, I mean, as you know, politics runs Congress, and they don't want someone telling the truth. I would be happy to, but they're not interested. Oh, Peak, one of our mods. Good to see you again. So, has anyone that's elderly got a new polio vaccine since the sugar cube back in the 60s? Probably not. Now, if you're going to a place where polio is endemic, if you don't have a vaccination record, CDC says you should get a new vaccination, which would be IPV. But if you have your record, as I do, no need to, no. Could there be a way to reproduce the old system where mothers make antibodies while an infection is induced? No, you could. Obviously, you could immunize the mother, and most mothers will be immunized against polio and will pass on antibodies to the kids. The problem is that they disappear after 6 months, right? And then the kids are susceptible. Whereas, uh, so the idea would be that the kids getting infected in the first few months, the mother's antibodies are there, and that'll protect them from paralytic disease. I don't know. It's a good question. I mean, that was natural infection. Maybe the vaccines don't do that, right? Maybe that's part of vaccine protection against paralysis is passive immunization from mothers to kids, at least with OPV. I don't know if IPV would do that. That's a good point. Yep. I assume it is impractical to vaccinate a newborn at birth when it has its mother. Yeah. There's for polio, you don't want to do it immediately. You want to wait. I think the first dose is a year to make sure the maternal antibodies are gone. And we just did a paper on TWIV a few weeks ago where they figured out when's the best time to vaccinate against measles, because again, you have maternal antibodies in the kid. There's a trade-off between waiting till the antibodies are gone and not getting infected, right? So they tried to figure out when that would be. Yeah. Elvis getting his polio shot on the Ed Sullivan Show meant a lot of kids were saved. Yep. So what's the equivalent of the Ed Sullivan Show today, where a celebrity could go get a COVID vaccine or some other vaccine, right? So would it matter? Is there a show that everyone watch? I have no idea. I don't know if there's no more mainstream TV. I have no idea. I just know that they're streaming. David said, "Did the cube come dosed, or did MD dose a blank cube?" I don't know. It's a good question. I think they dosed a blank cube. They would have these paper cups. They'd put a sugar cube in, and then they'd have a dose of vaccine in a vial and squeeze it on it. Yeah. I tried Paxlovid last week, but it made my symptoms, nausea and vomiting, worse, so I stopped it after 3 doses. Now, Red Hen, you shouldn't have done that because you don't know if the symptoms were due to Paxlovid or to the COVID getting worse. So I'm sorry you stopped it, but it seems like you're okay, right? Mark writes, if a child had oral vaccine in 1963, is he or she protected today? Yeah, if you do a lot of global travel, no, well, if you have a vaccine record, as I said, you don't need to. That child is protected. If you don't have a vaccine record, then you should get immunized just to be sure. Hmm. How did the vaccine that is attributed to vaccine-derived polio receive approval from regulators? Who dropped the ball? Well, that's a very interesting story, Rio. So as you know, IPV was licensed in 55, was used in the U.S. Sabin's OPV he developed those in the 50s, and he said to the U.S., I want to do a clinical trial of OPV. And they said, nope, we have IPV. We don't need your OPV. So Sabin went to the Soviet Union, where he knew Chumakov, who was the head of a very famous virologist, let's say. And he gave him his 3 strains of OPV, and they did a huge clinical trial of 100 million, 130 million kids, and there was excellent protection against paralysis. There was supposedly no adverse effects of the vaccine. So Sabin came back to the U.S. with those data. He showed the regulators that, okay, we'll license it. There was no evidence of VAP, vaccine-associated paralytic polio. But when the vaccine started to be used in the U.S., as you saw, in other countries, all of a sudden we're getting vaccine-associated polio at a rate of 1 in 1.5 million vaccine doses. So I don't know why they didn't see them in the Soviet Union. When I interviewed Kostya Chumakov, who was the son of Chumakov, who ran that trial in the Soviet Union with Albert Sabin, he said there might have been some polio in the vaccine arm of the trial, right? You wouldn't expect 0, but you wouldn't know if they were wild polio or vaccine, because we didn't have the methods for distinguishing that in the 1950s. So I don't think anyone dropped the ball. We just didn't have the data. Sally asks, what is the difference between making flu vaccine for a current variant and making a COVID vaccine for a current variant? Though some said it doesn't make sense for COVID. If so, why not? Well, so for flu, you figure out what's the current variant that's circulating. There are many ways to do that, but we won't get into it. And then you make a flu vaccine that matches the circulating flu variant. And that's what's been done for COVID a couple of times now. We had the original virus, which then went through multiple variants. And then we decided to make a couple of different Omicron-based variant vaccines. Same idea. You look at what's circulating, although for flu, there's way more data. There's a global network of people who look at whether the current viruses are evading the current vaccine or not. There's very little data for COVID. Daniel and I are always bemoaning this on the clinical update, that the CDC says we recommend this new booster, whatever the letters are now, in the fall, but the data are sparse, very sparse, nowhere near what we have for influenza virus. And so I'm beginning to wonder if, so they say, oh, there's increasing severe disease when the variant changes. That's why we want to match the vaccine to the variant for COVID. I don't know if it's because of the variant or because the immunity given by the mRNA vaccines are not long-lived. That's something that should be addressed. And there's some indications that the protein-based vaccines like Novavax could confer longer immunity, but no one has done those studies. So I have a suspicion that we don't need to change for COVID, the vaccine, to match the variant every year. We have to figure out how to make better, durable immunity. which may not be a feature of mRNA vaccines. Okay. Does that 1.5% solution of hydrogen peroxide destroy all viruses when rubbed on hands for 20 seconds? Oh my gosh. We never used to use hydrogen peroxide. Yeah, it will inactivate many viruses, right? There's one study of SARS-CoV-2, 3% will inactivate it in 30 seconds. There are literature reviews. Hydrogen peroxide can be gargled. It can be used as a nasal spray. On your hands. Let's see. Rubbing alcohol versus hydrogen peroxide. It says you should only use hydrogen peroxide on surfaces, not your skin, right? Because the peroxide can be, well, it's an oxidant, right? Hydrogen peroxide is too harsh to be used on skin. But although I don't know that the concentration you suggest would be a problem, but it may not destroy all the. So it can destroy many viruses and bacteria. Okay, that's what I know. Lori had IPV 2 doses in 56. Also had 2 sugar cube in the early 60s. I still have my vaccine cards. Yeah, so you have what I did. You got IPV and OPV. Peak said, seeing all you guys recounting your memories of OPV is making me feel so young. I got my IPV in 2005 or so. In any society, there are young people, there are intermediate age people, and there are old people. That's the way it is. I used to be young. And I couldn't stop the march of time. Lise says, I got both oral and IM polio vaccines, and my mom kept a list of dates with every vaccine. I still have the list. That's great. Wonderful. Rima got oral OPV and BCG. Do you know about the spike H vaccine nasal spray for COVID? The new vaccine relies on a protein scaffold that resembles a wire frame soccer ball. Yeah, like a buckyball. I think that's what we talked about it last summer. ASV was it, or the summer before? Virologist from Caltech. Let's see. That's her name. Pamela Bjorkman. She was working on one of those. It was very interesting. Yes. I haven't heard since she talked about it. It was a science paper originally where she published the first results obtained with it. Yeah. Spamlet, I'm younger than you, but I had to have 3 injections. My brother ran away from the clinic and was rewarded with the sugar cubes. You had 3 serotypes. No, 3 serotypes, each in a separate cube. In 1960, whatever that was, 1963-4, they were doing monovalent OPV, and then they realized, the rate of VAP was unacceptable. It was unacceptable, so they combined them. Yeah. If Tryon says, if after IPV we drink OPV after 30 days. Yeah, that is one solution to preventing VAP. Yes, but you still introduce OPV into the environment, so I don't think it's a good idea. Is that why tetanus hurts so much? Because of the inflammation? Yeah, tetanus vaccine is a toxoid. an inactivated toxoid. Um, I'm trying to see if it's adjuvanted. But if it hurts, whether or not it's adjuvanted, the hurting is inflammation. Yeah. Speaking of adjuvants, I wonder how efficient designed protein adjuvants would be. That would be something for the explore. Yeah, the used adjuvants now are not proteins, right? They're lipids like the lipid from bacterial cell walls, and also oil and water mixtures, squalene. So it would be interesting to make protein-based adjuvants. You could certainly figure out what would induce inflammation. Yeah. Smallpox vaccine hurt and left a scar. Yes, I have my smallpox vaccine scar, because what they do is they take a needle and they scrape the surface of your skin. And the needle has a drop of vaccine in it. They scrape it, and then those cells get infected and makes a pustule. Yeah, it hurts. And then you're left with a scar. Yep. 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 Maybe mRNA vaccination dreamed to eradicate some viruses. I don't think so. I think as I said, I think the immunity is not sufficiently long-lived to eradicate viruses. I think protein-based are the ways to go. Who the hell is making up these rules regarding polio research? WHO, CDC, Global Polio Eradication Initiative. Then there's some other organizations that are involved in the containment, and most of them are epidemiologists and bureaucrats who don't know any poliovirus biology. Ken, thank you for your support of Microbe TV Science Communication. We really appreciate it. Sounds like I should look into IPV with my doc. I have no vaccine record. No, nothing would show up. So I think you should talk to your doc about getting another one. Yeah. If you're going to go somewhere. Your gut is a gain of function laboratory. It sure is. Oh, look at the Philip Roth novel Nemesis is set in summer 1944 Newark during a polio epidemic. It reads like an autobiography mixed with a suspense thriller. Oh, I have to check that out. Thank you. By the way, folks, how many famous people do you know had polio besides FDR? Let's see, Nemesis. Philip. So I can think of Itzhak Perlman and Neil Young. Can you name some others? Famous people who had polio. After my second smallpox vaccine didn't take, doctor told mom I had natural immunity. Oh, okay. I'm not sure they would know if it took unless they did an antibody titer, but it's okay. Auntie got a dengue warning today for Long Island. Yeah, so there have been about 150 cases in New York and New Jersey. Apparently, all people who acquired it in travel, right? This is the travel season. There are a lot of dengue cases in Puerto Rico, but also other countries have a lot of dengue as well. Hmm. I'm seeing news reports that dengue might follow the hurricane. Yeah, but so far all these cases are travel associated, according to CDC. Can the metaphor of type of vehicle used be compared to gain of function virus on bicycle or taxi? No, I don't know. I don't understand. Sorry. I don't understand. I'm thick. Any guesses as to why mRNA vaccine might give shorter-lived immunity? I don't know. I think maybe with the protein-based, you're getting a lot more protein injected, and that maybe the mRNA vaccines don't make as much, but I'm not sure. I'm not sure. Yeah, don't drink hydrogen peroxide. John says a 1.5% solution of hydrogen peroxide only dries the skin, which can be corrected with moisturizers. Otherwise harmless. Well, I don't want to give any cleansing advice here with hydrogen peroxide, so be careful, please. Mark uses a 3% hydrogen peroxide to sterilize wine barrels when the bung is removed for topping or bottling. Yeah, wine barrel, that's different, right? Yeah. Vanity gives her dog hydrogen peroxide to make him vomit. Vincent and colleagues, biomedical researchers, are heroes to my mom who endured paralytic polio at 51. Now at age 99 plus, post-polio is tougher. I'm sorry, Tom. Really sorry about that. But 99 plus is pretty good, right? When I was a kid, H2O2 was used as a wound disinfectant. Recently, I was told it's a bad idea because it destroys cells that are needed for skin repair. So I don't know anything about this. I used to not be able to move my arm after a tetanus vaccine until I discovered that if I kept moving my arm for hours, my arm would not go through paralysis. Okay. All right. Who else got polio? Joni Mitchell. Yeah. Neil Young. Helpless. Yep. Helpless, 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 helpless. Does anyone remember getting a cube with 3 colored stripes? Maybe OPV 1, 2, and 3. I've never heard that. But I didn't. Ooh, look, Amy is here. Alan Alda had polio. Robert Redford. Mitch McConnell. That's right. Excellent. No rules by CDC. Rules for what did I say? All right, whatever I said that the CDC was making rules for is wrong. If I had adjuvant squalene, will I be immune to sharks? Nope. You won't be immune to sharks. Yes, I'm listening. I just listened to you say no rules by CDC. Yes, eradication is dictated by the World Health Assembly, not CDC. My first polio victim comes to mind is Ian Dury, author of Sex and Drugs and Rock and Roll. Artemis is Arthur C. Clarke. Amy says the mRNA vaccines don't induce interferon, right? They were designed not to. And so that may be why they don't make as durable immunity. Thank you, Amy. That's really good. Really good. More famous people who had polio: Mia Farrow, Alan Alda, Francis Ford Coppola, Donald Sutherland. Yeah, how about that? Amy. Okay. A virus rides a slower bike through busy traffic of taxis, or taxis carry more virus faster for specificity. You wanted me to know if that's what an adjuvant does. Let me go back and look at your original question because I don't even remember it now. Can the metaphor of type of vehicle use be compared to gain of function? So, yeah, why not? Faster, right? It could be. Amy Costello wants to know, how's our plaque assay princess been? All right, that's the end of our questions. We had 173 people. There's one more question here from Visto. Could poliovirus surface proteins be used as an oral vaccine? No. No, I don't. That just you can't. Maybe you could make an empty particle, right? People are working on what are called virus-like particles, but they would have to be injected. Yes, you can make proteins in GMO plants for sure. Definitely. Wayne remembers Alan Dove explaining that adjuvants piss off the immune system. Yeah, that's really good. I like that. Margot wants to know why the mRNA vaccines. Amy says she's busy in response to Questella. Margot, why don't they induce interferon, the mRNA vaccines? Because the bases have been modified to not induce interferon. Otherwise, they're cleared. The mRNA vaccines are cleared if you just use the wild type bases, the normal chemical A, C, G, U, right? So that's one of the things that Kathleen Carrico did years ago, is figure out what base to change to make the mRNA vaccines not induce interferon, so that the RNA stays around and is translated into protein. But that may be why not inducing interferon is really important for a good adaptive response. So the protein-based vaccine will induce interferon. Okay. Can you talk about the different shingles vaccines? So there's a chickenpox vaccine for kids, which is an attenuated infectious virus, right? And the shingles vaccine used to be a higher dose version of that, but a new one was made called Shingrix, which is a viral protein mixed with adjuvant, which is great. It really is great at preventing shingles, and they're actually thinking of replacing the kids' chickenpox vaccine with Shingrix, maybe a lower dose. Now, what was I going to say? If you already had shingles, you should get Shingrix, yes, because you could get it again, and Shingrix would prevent that. Yes, yes, yes. If politicians knew their favorite beer is a result of gain of function, maybe they'll leave it alone. Oh, yeah. And Amy says virus-like particles are not stable enough. Yeah, so poliovirus-like particles, which would be non-infectious, injected, easy to make, are not stable enough. And she also says they're not also appropriately immunogenic. So why would a VLP be better than IPV? Well, then you have to grow up infectious virus, right? Which is a containment issue, perhaps in its own. I thought the durability of a vaccine has more to do with how quickly the germ mutates. No, not at all. It has to do. I mean, if influenza virus changes, yeah, then the durability is gone. But other viruses do not change that way, and you can either get durable immunity or not. And one of the ideas that mucosal infections do not generally provide good durability, and viruses that cause viremias do. Yeah, but we're not clear on the details. But it's not just the mutation of the virus; it is how the vaccine interacts with the immune system. Why do you think Moderna and BioNTech were successful, but CureVax wasn't? I don't know. Did they try? Yeah, the CureVac. They have a COVID vaccine. Yeah. I mean, they are marketing it, right, in a number of other countries because because Moderna and Pfizer couldn't service everyone, so there's a market for CureVac. I don't know the specifics though. I couldn't tell you. Some adjuvants increase the persistence of antigen. That's right. I left that out for simplicity, but some antigens concentrate the antigen and have it released slowly from the inoculation site, so it persists longer. Yes, you're absolutely right about that. And Amy says, yes, a virus for IPV is a containment issue. So after containment kicks in next year, well, the manufacturers of IPV will have to be certified as able to work with poliovirus, which is a pain. And so this would get around it. Yeah. All right, it's 9:59. Time to wrap up this puppy. Let me thank all of our moderators today. We had Vanity Nutrition. We had Andrew. We had Tom. We had Peak. We had very briefly at the beginning. We had Steph. Very briefly, we had Tom. Did I get everyone? I believe so. Thank you all for moderating tonight and helping to make this civilized and also participating in the conversation. And I want to thank all of you 174 people for joining us this evening. Our next live stream will be next Wednesday. That's July 17th, 8 p.m. Eastern Time. Thank you, Amy, for coming and participating and answering questions. Love it. And thank you all for being here, folks. Have a good evening. And until next week, be safe. Bye-bye.